All right. Hello, everyone. Today we're back at a decent time. We're not after midnight, uh, and I think today we're probably going to be doing some um, maybe. I think we're going to be doing like biochem. So I know. I think I literally just said yesterday. It's kind of ironic. I literally just said yesterday that we weren't going to be doing biochemistry or biology for a while, and then here I am. But it just needed to happen. So, um, yeah, I think honestly, I want to do it like we'll see how long it takes me to do these cards because the enzyme section is almost completely this section here is almost completely the complete wow, I'm studying a lot. The whole chapter is basically these 50 terms. There's like one term in addition to this. Watch, I'm gonna set this up to like 60. Honestly, I'll set it up to 75. I'll set 75. So, yeah, so, okay. All right. I thought that I was confusing it with chapter four. Okay. So, yeah, this whole chapter is pretty much 60 terms. I might do the remaining like 15 and maybe psych social. Uh, because psych social, the next part, as I told you guys, is like, it's just too much. It's just a lot of work, a lot of memorization, a lot of lists. Well, obviously this is all memorization, but like it's a lot of lists. So, it's gonna make it a lot more painful. 126 terms in Mr. Pancott of the 150. Okay. I guess that makes sense. We've been doing that a lot the last couple days. All right. Well, let's get it. Okay. Well, can I do that? No. It's gonna start me up here. Um, yeah. Let's put this down to 0 again, per usual. I have this. I feel like it's possible that someone in the future might say like, why do I always set it to 0? The answer is that although I already have the setting enabled where reviews go first, I'd rather just have that like control where it like I'm not gonna slip into the new cards. I just don't want to do that. Theta waves are slower. Oh, sorry, forgot about the lo-fi. Let's see. Okay, add a good volume. We'll put it up. Okay. Theta waves are slower. Okay, hold on. Theta waves are slower slash lower frequency waves that are associated with um theta waves are slower slash lower frequency waves. Uh Drowsiness and right after falling asleep to push fluid through the coffee from N to C. Um the steric theory of olfaction or shape theory. Goose Deucin. Labeled line model. Latent content. N1 and 2 and 3 and 2 REM N1. Behavior slash conscious. You know, if I move like this is what will happen. The olfactory bulb. K complexes. I don't even know how is that a K. Like, how do you get a K from that? Like, this is a K complex. How is this? Where do you? Why is it not called like a Z complex? Like that. Even that's more. understandable. Where in the world do you get a K from? Hypnic jerk, consciousness. What do roles allow us to ascertain? The energy level of the valence electrons, beta waves, oval window, daydreaming. Oh, this card hasn't been shown in a while. Um, C equals wavelength times frequency, form, alpha. The special part of retina rich in cones, the macula. Um, rods detect light and discern low level of detail. Okay, so for olfaction, so a molecule binds to the GPCR on the olfactory epithelium. The G protein goes and opens ion channels. The ion channels lead to the opening of the ion channels uh, leads to an action potential that goes through the cribriform plate to the olfactory glomeruli, to the metrotufted cells, to the and those synapses in the brain. Nice. Just a second bond. Contextual effects, circadian rhythm. Say yes. Dreams are threat simulations to prepare for the real world. To bulge out as fluid as fluid as fluid flows through the cochlea. Gustatory cortex, anterior chamber, kinesthesia, tetrahedral, more paradoxical sleep, the stirrup, 20 hertz and 20 kilohertz. What is denaturation? It is like the loss of a protein's shape uh, due to like external factors like heat, detergent, or solutes. I mean, yeah. I just thing is right. I don't need to say by disrupting bronze. Like the strategy might result in some. If you always say yes, you're gonna get all the hits, but you're also gonna have some false alarms. Olfactory bulb separates the piriform plate separates the um separates the olfactory epithelium from the brain. Three types of taste buds are foliate, fungiform, and circumvallate. Pineal gland, sleep spindles, hypnagogic hallucinations. 1/2000th of a proton. The manifest content. Damn, this one hasn't been shown in a while either. So basically, you have the DNA sample, you split it into four different tubes, and then you're gonna run it through a PCR with DDNTPs as part of the like solution, as well as the NTPs. And basically, because the DDNTPs lack a 3' hydroxyl group, it prevents elongation from that side. And so by ordering by putting them through gel electrophoresis afterward, because the DDNTPs would have bound in different places, and the four different tubes have different DDNTPs. So one tube might have DDATP, another one DDCTP. Like they're all gonna be they're gonna stop elongation at different points. So it makes for different length profiles, and the gel electrophoresis would be able to catch that. And the different levels would basically tell you, as long as you order them from the ones that travel the furthest to the ones that travel the least far, you're gonna get the exact um, gene or nucleotide order or code, I guess. Uh, what salty and sour. The primary auditory cortex is part of the temporal. And three. What does a magnetic quantum number tell you? It tells you the the shape. Like, it doesn't tell you the shape. It tells you the orientation of the subshell. It's called non-REM. The fourth stage is REM. Them are connected by tip links. It's a change or subject to sensory adaptation. Cilia in the ear are called hair bundles. Dreams are, it's just the brain interpreting random neural impulses from the brain stem or pons. During N1 sleep, 90 minutes, is from the pinna to the tympanic membrane. Is olfaction. The state just before falling asleep or waking up, drowsiness. The sclera contains alpha, beta, and desynchronous waves. To maintain pressure in the front of the eye and to provide nutrients to the iris/cornea. Sends sensory signals to mitral/septal cells. Uh, the olfactory glomeruli. It's to describe the position of an electron. When tip links are pushed back, so the K plus channel is open, the Ca2 plus channel is open. Then that's going to cause an action potential that's going to travel to the spiral ganglion cells, then to the going to travel to the so okay, my fault. So yeah, so it's going to send the action potential to the spiral ganglion cell, and then to the auditory nerve, and then to the brain. that each it kind of makes sense of it's like hearing has a spiral ganglion cell, and like that's like I th- I'm, I feel like there's the semicircular canals that kind of starts to form a spiral. So it's kind of like intuitive. States that each sensory receptor binds to a specific stimuli is directly linked to the brain. Um, um, this is the label line theory, vibrational theory of olfaction, alpha, beta, delta, theta. Will take confirmation and eventually lead to an action potential for memory consolidation and forming episodic memories. Melatonin, activation synthesis hypothesis, linear. Um, it it releases energy. Anterior chamber is filled with aqueous humor. Its secondary structure with its characteristic bond. Um, secondary structure. I don't know about the definition. Like, it's just like I feel like I must have never really gave a definition for this. Secondary structure is it's like these are like beta sheets and alpha helices that form due to like hydrogen bonds between the like in the backbone. Its characteristic bond. I just said it. Um, oh, you see, here's what I'm saying. Like it's more like I'm gonna edit the question because it's gonna cause me to get it wrong because I don't know what the question. I didn't know that that's. I would have known that that's all it was asking. I wouldn't have had that tough of a time. Three phenomena that can occur during anyone's sleep are um, hypnagogic hallucinations, Tetris effect, and hypnojerks. Consciousness ranges from alert to asleep. Uh, I'm gonna give myself good on that because there's no way I forget that. Slower, lower frequency waves that are associated with drowsiness and right after falling asleep. I also want you guys to know, I don't know if I said this before, but if I just skip through a card, like especially on a review, if I skip through a card, I don't know if y'all would really even care or mind or want to know, but I'm pretty much pressing hard like every time. There's there's like a 0% chance that I click anything but hard. Like I have to know it like really well. Like it didn't even take any mental effort for me to pull that out of my head. Um, it has to take like basically like negative effort for for me to click good. Um, and I press easy if I could have answered that question in my sleep. Are slower lower frequency that are associated. Didn't I just do this? Like this is like my first card. Drowsy. alpha, not alpha. It's not alpha. It's not. It's theta. How is that 3 days? I put 3 days. How's that 3 days? What is a conjugated protein? Um, it's a protein that derives a part of its function from a covalently bonded prosthetic group. Okay, phototransduction cascade. All right, let's see if I still got this. This low key, I thought it was gonna show up a lot more. I don't know what I clicked, but it I don't know. So okay, so basically, it's gonna be a protein binds. Wait, hold up. Wait, wait a second. Okay, so light hits, not a protein. A light hits cisretinol. The cisretinol it changes conformation into transretinol, and um that causes the release of that causes wait, is it yeah. That causes rhodopsin to change shape, and rhodopsin changing shape causes transducin to be released, and transducin goes and activates phosphodiesterase, and phosphodiesterase is gonna change all I think it's CGMPs to just GMPs, and the lack of CGMPs causes Na plus channels to close, and it causes hyperpolarization of the cell, and the rod cell turns off. Thank God. The CGMP and the GMP, those are kind of hard to remember. Um, Serine, threonine, and tyrosine. Consciousness is the awareness of our self and environment. Pulls on suspensory ligaments to change the shape of the lens. Um, Ciliary muscle uses background knowledge driven by theory. Inner ear includes semicircular canals and cochlea. Amplification, place theory. Um, no, yeah, mesopic. They rely on GPCR. Begins with the data. Oh man, it's called paradoxical sleep because um, we have similar brain activity in our sleep, uh, like as we do when we're awake. A Twitter ion is perfectly neutral because its charges cancel out. The TRPV1. I would be really screwed if they spoke about the other TRPs, because once we get into like the TRP A channel, like bro, I don't. That's when I really start. That's when I really start like dying inside. Um, in GTA. Is a transparent thick of sheet of fibrous tissue that permanently cornea. Electrical active transmission. Theta waves, K complexes, and sleep spindles. Cones, two nonmetals. Theta waves. At the base are high frequency. K complexes suppress cortical arousal and aid in encoding declarative/explicit memories. I kind of said that, but I didn't say that to keep you asleep part. So I'm gonna give myself again. What is the effective nuclear charge? It is the attraction felt by the valence electrons despite the shielding electrons. It's found Zeph is equal to protons minus shielding electrons. Valine is just boom. You see, now we have. Oh no, yeah. So valine is like this. I was like, damn, this is too similar to threonine, but threonine has the OH there. That's like the difference. Olfactory bulb is an extension from the brain that consists of a bundle of nerve fibers that send little projections through the cribriform plate to the olfactory epithelium. Delta waves are associated with deep sleep and comas. I realized I was saying that, but before the task was complete, I did click the button. Anyways, and I didn't look up, but I'm gonna say that I would have gotten that. Taste, rely on GPCRs. Sweet, bitter, and umami. It's a bond that forms between the amino and the carboxylic acid in amino acids. N2, as principal energy level goes up, it increases. Dipole, focus slash concentration, daydreaming, somatosensory homunculus, organ of Cordy, um, inside back area of the eye filled with photoreceptors, electroencephalograms, olfaction, Tetris effect. Say no unless 100% certain that the signal is present. Also, aqueous humor, cochlea to differentiate between sounds. Basilar tuning, the phobia, A delta, serious system, sensitive. Head positioning and actually no balance and damn. I'm gonna say I think it's balance and spatial orientation. What are aspartic acids? Aspartic acid is gonna be, I think it's D. Is gonna be the one letter abbreviation. It's ASP. Its character obviously acidic. I wasn't gonna draw it out because I know it. Organ of Cordy includes the basilar and tectorial membrane. Sensory narrow hearing loss. Wait, guys, give me 1 second. I'll be back. I'm back, guys. Um, so Sigmund Freud stated that dreams are thoughts and desires that need to be interpreted. Okay. Bottom-up processing begins with the stimulus. More susceptible to poor attention, poor memory, obesity, depression, glomerulus, gate control theory, the neurons protecting from olfactory chemoreceptors, sweet, sour, salty, bitter, umami, filiform babble, um, to inhibit certain perceptions and maintain a tranquil state, suppress cortical arousal to keep you asleep, and aid in encode aid in encoding declarative slash explicit memory, is associated with slow wave sleep, delta waves, uh, sleepwalking slash sleep talking, and declarative memory storage. Rich in cones, prefrontal cortex, choroid, accessory olfactory bulb, uh, sleep, bursts of brain activity, olfactory epithelium. The blank separates the olfactory epithelium from the brain. Um, the cribriform plate, delta waves, glutamate are unconscious thoughts and desires that need to be interpreted. All right. Now we can put it at 75, and we're gonna try to see if we can finish. It took us a while, low key, to get through the reviews today. Um, I might average maybe like if I do 6 days. Damn, that's not enough. We need so if we did 50 divided by 6, that's like 8 or 9. So maybe every day I might do like 59 terms, something like that. Like 59 terms, just to negate the to negate um the 50 term day that I miss. Instead of doing 75, I realize I'm not cutting out a lot, and I'm making it more like more of a constant thing. But I think it's better than doing it all in one day. I might just make it an even 60, to be honest with you. Um, and also finish the deck faster. Okay, so if we do 75, if we're doing 75 per day, when does it say? When does it say I'll be done with the deck? 69 days. So it basically thinks I'll be done by September 3rd. At this rate, I'll be done by September 3rd. And then okay. So uh yeah, we're gonna go with enzymes, I think. Oh my god. This deck is so annoying, but I know that it's annoying because I need to do the fill in the blanks. An exergonic reaction is a reaction that releases energy with a negative delta G. Um, I need to look up this definition. I feel like, I think that definition is wrong. It's more like it's negative delta H. All right. What is a hollow enzyme? Uh, I don't know. I don't remember. Is an enzyme with its necessary cofactor? Okay. What is uncompetitive inhibition? Inhibitor binds to the enzyme substrate complex and locks it up, preventing the release of products. Let me look through the cards to see if okay. Anyways, let me look through the cards to see if they're gonna give me the graph as a card. I don't think they do. Oh, never mind. I'm bugging. I think I was just going too fast. Yeah, I was going too fast. Okay. It is a reaction with a negative delta H, and that releases energy. What is a hollow enzyme? It's an enzyme attached to its necessary cofactor. What is an isomerase? An enzyme that turns a molecule into its isomer. Do they ask me what an isomer is? Oh, okay. They're gonna teach me I'm gonna learn what isomers are in one of the other chapters, so it's okay. Catalyze the rearrangement of bonds within a molecule. Um, uncompetitive inhibitors. It's where uncompetitive inhibitors bind to the. Okay, hold up. Let's lower this a little bit. Um, okay, so this is where uncompetitive inhibitors are going to bind to the enzyme substrate complex and lock it up so it can't release the products. Michaelis-Menten equation. I forgot. Oh. I don't like that order, but it'll help me remember. Um okay. So what is an isomerase? An isomerase is an enzyme that catalyzes the rearrangement of bonds within a molecule. It also aids in reactions between stereoisomers and constitutional isomers. What is the slope of a Line-Weaver-Burk plot? So slope is equal to y2 minus y1 over x2 minus x1. If we take this point and this point, this one's gonna be, we can take this one as the first one, this one as the second one. So the x is gonna be 0. So, and then the numerator is gonna be 1 over v max. This can also be represented as v max to the negative first. And then this one is gonna have a y of 0 and an x of negative 1 divided by km. So what this ends up doing is that we get v max to the negative first over km to the negative first. And then these essentially are just going to flip. They're going to km over v max. Okay. I just need to like have that in my head. That's like pretty basic stuff, but as long as I have that in my head, it'll make it easier for me not to forget. I might still forget it, of course, but Michaelis-Menten equation is going to be V naught is equal to V max. I think it's minus the concentration of S divided by the concentration of S plus Km. Oh, damn, it's multiplied. Damn. A reaction with it's a reaction with negative delta H and that releases energy. What is a hollow enzyme? What is a hollow enzyme? We're gonna reset this just because I genuinely want to see how long this is gonna take me. This is the reason why I've been delaying this because it's gonna take forever. What is a hollow enzyme? It is a protein that it's a protein that is attached to its necessary cofactor. What is uncompetitive inhibition? It is when an inhibitor binds to the enzyme substrate complex and locks it up, preventing products from being released. What is the slope of a line? It's going to be Km over V max. This is going to be V naught is equal to V max times concentration of S divided by concentration of S plus Km. What is a lyase? Uh, I don't know. Um, Catalyzes the cleavage of a single molecule into two products without addition of H2O. Summarize. Um, Damn, 214 minutes now. Oh, brother. An isomerase is um an isomerase is an enzyme that catalyzes the rearrangement of molecules and rearrangement of bonds in a molecule. Let me add the second part. So okay, I needed something else to trigger me saying what the note says. A lyase uh, separates a molecule into two products, catalyzes the cleavage. Six features of enzymes: lowers activation energy, increase reaction rate, not changed or consumed, very sensitive to the environment, do not alter delta G or the equilibrium constant, and are very specific. Damn, guys, my fault. Hold up again. Okay. All right, guys. Start again. Um. Okay. A hydrolase catalyzes cleavage with the addition of water. Summarize. In a summarize, catalyzes the reorganization of bonds within a molecule. It also catalyzes reactions between stereoisomers and constitutional isomers. It catalyzes the cleavage of a single molecule into two products without the addition of water. Increase activation energy. Um, it's very specific. It's um, it doesn't affect the. Oh, it's not consumed or not changed, whatever. Um, I think that's all I got. Increased reaction rate. Doesn't alter delta G or the equilibrium constant. I thought about very sensitive for literally like a millisecond. I just couldn't catch the thought. It's like like flew by my head in a plane. What is the slope of a line? It's Km divided by Vmax. Michaelis-Menten equation. V naught is equal to Vmax times the concentration of S divided by the concentration of S plus Km. What is a holoenzyme? It's an enzyme bound to its necessary cofactor. What is an exergonic reaction? It's a reaction with a negative delta H and that releases energy. It catalyzes the cleavage of a single molecule into two products using hydrolysis or with the addition of water. What is uncompetitive inhibition? It's when the inhibitor is gonna bind to the enzyme substrate complex and prevent it from and lock it in so it it can't release the products. What are the six key features of enzymes? Um, okay, so it decreases. I don't know why I said increases last time. Decreases the activation energy. It increases the reaction rate. It's very sensitive to the environment. Um, it's not consumed. It doesn't alter the delta G or the equilibrium constant, and it's very specific. What is feedback regulation? Feedback regulation is like, it means that if a protein is um what's it called? If a protein has okay, in a specific pathway, you might have a product and you want a certain amount of that product. So a feedback regulation is, it means that those end products or any intermediates along the way might be able to come back to one of the proteins that led to it, not necessarily the protein that or the enzyme that created it, but one of the enzymes in the pathways leading up to it, in the pathway leading up to it, might be able to go back and inhibit the protein from continuing to make it because the cell might be producing, so that the cell doesn't waste resources. Yeah, I feel like I have this down. Like I know what it is. I can describe it. What is an Apple enzyme? It's an enzyme that is not bound to its cofactor. The y-intercept of the Lineweaver-Burke plot gives us the Vmax, or 1 over Vmax. So how does an uncompetitive inhibitor affect the Vmax Km? Okay, so let's think this through. So if we have an inhibitor that is going to, if we have an inhibitor that binds to the enzyme substrate complex and it prevents the products from being released, that's only after it's made. So Km, this is the affinity that a sub like that an enzyme has for its substrate, I'm pretty sure. The Vmax is obviously like the velocity in making it. If the uncompetitive inhibitor is binding to the enzyme substrate complex, then the Vmax technically, I feel like it shouldn't be changed. The Km, but the problem arises when you consider what happens after. After it's bound, the Vmax is obviously going to decrease fundamentally to 0 because it's not going to make any more products. The affinity that it has for the affinity that the enzyme has for its substrate, its substrate, is essentially going to increase like crazy, right? Or I think it would have to, I think it has to increase. But the thing is that it's the enzyme substrate complex and it's going to stay as the enzyme substrate complex unless it's more like bound to the EP, the enzyme product complex or something like that. But so I feel like we can determine what happens to the Lineweaver plot after we figure out what the Vmax and Km do. They increase affinity, lower Km. So Vmax decreases and Km also decreases. Okay, my problem is that a lower Km is equal to a higher affinity. That was part of the problem. Um, um, Okay. So, okay, so as shown here, it's -1/km. So since it's going to be -1/km, and we know that the km is going down, when km goes down, it's -1/km. If it goes down, 1/, it's like 1/4 versus 1/3. So yeah, so you're gonna keep moving that way. Um, and then you're moved that way because the cam was lowered, and the Vmax is gonna decrease as well. So it just gets further. They both get further from zero. It gets further from zero in both directions because it's a one because it's 1/. So it just gets they both get further from zero, and so it just the whole line just becomes parallel, and like and more of the line ends up in the fourth quadrant, or not the fourth quadrant, right? What quadrant is that? Is it the second? Okay, so all right, that should help a little bit. The y-intercept of the Lineweaver-Burke plot gives us the y-intercept is 1/Vmax. What is an isomerase? An isomerase catalyzes the rearrangement of bonds in a molecule. It also catalyzes reactions between stereoisomers and constitutional isomers. The slope is Km divided by Vmax. It is hydrolase catalyzes the cleavage of a single molecule into two products using water. So this is going to increase or decrease activation energy, increase reaction rate, um, very sensitive to the environment, very specific. It's not consumed, and it doesn't alter the delta G or equilibrium constant. What is a lyase? It catalyzes the cleavage of a molecule into two products without water. Okay, let's mention equation. It's V naught is equal to V max times concentration of S divided by concentration of S plus Km. How does an uncompetitive inhibitor affect? It brings both of them further from zero. So it brings that one up and then brings that one that way. The V max is going to decrease and the Km is also going to decrease. What is the catalytic efficiency? Kcat. I'm not sure. I don't remember that formula. Catalytic efficiency. My one problem with this deck is that it breaks it down, but it's like it breaks it down into a random order. Like, why were these? How did they make? How is it that you reference things before you explain the definitions? KCAT divided by KM. Okay, these two, this whole, all of that, this is literally just explaining how math works in this. So I'm just like I'm not, I'm like, I feel like you kind of have to be able to figure that out. Like, I appreciate it for telling me what KCAT and KM are, but what is uncompetitive inhibition? It means that the inhibitor is going to bind to the enzyme substrate complex. Hello, guys. Yeah. Okay. What up? Whatever. So yeah, so it binds to the enzyme substrate complex, and it um it's like a real this is like really bad. It binds to the enzyme substrate complex, and It binds to the enzyme substrate complex. I'm like blanking. It binds to the enzyme substrate complex and locks it in, so the product can't be released. What is an apple enzyme? It's an enzyme without its necessary cofactor. It's Kcat divided by Km. Decrease the activation energy. Um, Increase the reaction rate. It's not consumed. It's very sensitive to the environment, very specific, and doesn't alter the delta G or the equilibrium constant. What is mixed inhibition? I don't remember at all, to be honest with you. Okay, so a competitive inhibitor is going to bind to the same site as the substrate. So I feel like Cam would stay the same because like competitive inhibitors, they get less effective the more substrate you have. So I feel like Cam would stay the same. The affinity of the enzyme for its substrate, I think, is the same. Vmax, what's going to happen to the Vmax? Um, uh, the Vmax would. I feel like the maximum rate with a competitive inhibitor would have to decrease. Like it has to decrease, right? Damn. Okay, so since you need more substrate, oh, that's fair because CAM is also half of the substrate concentration you need to reach half of Vmax, and you're gonna need more of that. You're gonna need more of the substrate to reach half of Vmax. So that does make sense. Um, and Vmax is the one that doesn't change because when the substrate does bind, it can still reach the same maximum velocity as you increase substrate. Yeah, that makes sense. And then so that means that so you need more substrate and you need your Vmax isn't going to change. So it's going to have the same, like if the normal line goes like this, like we're going to create, we're going to pretend like that's a, like this is a normal graph. If the normal line looks like that, then if Km is increasing, if Km is increasing, it's going to look like something like this. It's going to be like steeper. I promise I didn't look at this. I'm looking at this as a verification, but I didn't look at that at all. That was not like that was not me literally redrawing this. Um what is mixed inhibition? Uh Vmax is going to decrease. It means that it can bind to either the enzyme or the enzyme substrate complex. Um I guess okay, so let's take this true because I know that the Vmax is going to decrease regardless. But if okay, so Vmax is gonna decrease regardless. The Km can increase or decrease. So let's think this through. So if it binds to the enzyme, um, we know that if it binds to the enzyme at some like allosteric site, then the enzyme wouldn't even be able to take it in. And so it might act as it's not able to take in the substrate. So, or wouldn't be able to bind it. But like, I feel like, no. Yeah. So let's say it wasn't able to bind it. Uh, and so the V max, or like it loses affinity for for like if it loses affinity for its substrate, if it lost affinity for its substrate, then okay, let's look up what causes the effects. What causes the effects of mixed inhibition? Let me see if maybe like maybe Yusuf Hassan's videos can explain it. Um, all right. Um, so let's see. I'm gonna I'm gonna share this video. Let me see if I could put it in the All right. So I just fixed the description. I'm about to put the video so we can all watch the thing on mixed inhibition so we can kind of try to understand it. Let me see.
I know they're not parallel. That I'm not going to fix. So the inhibited line is parallel to the original line, shifted to the left. You guys get that? Pretend they're parallel. Just pretend. How much space do I have over here?
A little bit.
It's fine. This one's going to be short. Mixed inhibition. I apologize, everyone. Video cut like just at the last moment, so we just have a little bit left to go. We're talking about mixed inhibitors right now. It binds the enzyme. Give me that marker behind you. It binds the enzyme or the enzyme substrate complex. But the thing is, when it binds them, but with different affinity for each. So this is where, like, you remember how over here everything changed by the same amount, so the Km wasn't affected? This, like, there's no telling which way what's going to get affected, what way. Like, it's different for everything, you know what I mean? So with different affinity for each, right, it also binds to the allosteric site. Allosteric binding, right? If it had the same affinity for both of them, it would be the same as a non-competitive inhibitor. It has different affinity for them both. It binds the enzyme to some degree, binds the enzyme substrate complex to some degree, and maybe when it reforms, it binds this one more than the other in a certain moment, and blah, blah, and it's this weird sort of like push and pull, ping pong like mechanism. You know what I mean? And the thing is, if E is bound preferentially, this leads to an increase in the Km. Because you get less ES formation, right? Same thing as before. And if ES is bound preferentially, you get a lower Km, the same way you did over here, right? And in both cases, Vmax drops. And the Line-Weaver-Burke plots intersect at a point not on the axis. You know why we put this statement here? Do you guys know why we put that there? It means you don't have to draw it. You don't have to know what it looks like. Because you'll never be able to tell where they cross.
Would they just be dependent on what it does if we just have to look at the specific inhibitor?
Sorry, what did you say?
Like, would you just have to look at the specific mixed inhibitor? There's not a graph for mixed inhibition?
Yeah, you'd have to look at the specific inhibitor and find out exactly what it's doing. Yeah. Take a photo. This was the way I wanted this lecture to go. I think we really, really got through this one well. I think you guys were very patient. Good job, guys. Take a screenshot if you're at home. Rewatch the video. Take your notes. Let's get through the final half page of notes for the lecture. And then we'll go home. I know you guys are tired.
Okay, so let's go back.
From the perspective.
Like non-competitive and the uncompetitive.
Hey Meta, Cool Claire. Hello. I do have some gossip to tell you. Are you still in New York?
With Sours by Extra, every chew is sour.
Dude, it's been 3 hours. It's an allosteric activator. The inhibitor binds to an allosteric site. We've seen allosteric before. Where? Where's the very famous example of allosteric activation? Huh? 10 o'clock?
You can file a noise complaint, I think, at 10 o'clock.
Someone go file a noise complaint. What's the very famous example of allosteric activation? Someone look this up. I'm pretty sure fructose 2,6-bisphosphate is an allosteric activator of PFK1. I'm pretty sure. I believe it's allosteric. This is allosteric inhibition. It binds to an allosteric site. So the allosteric site is a site different than the active site, but binding at that site changes the conformation of the enzyme enough that the active site gets affected. Does that make sense? So this leads to a change in conformation of the enzyme. And remember that conformation is just another word for shape. Change in the shape of the enzyme. This is where the English word conform comes from, to conform, to change your shape to mend to something.
How would that work with something like PFK2 where it's like positively impacting?
Yeah, like fructose 2,6-bisphosphate. So fructose 2,6-bisphosphate, if it's an allosteric activator, I'm not sure. It is? OK, perfect. Fructose 2,6-bisphosphate binds to PFK1 and changes the active site in a way that better accommodates fructose 6-phosphate. Right? It changes the active site in a way that actually accommodates it nicer, which actually gives a little bit of premise to the induced fit theory. Doesn't that make sense? That you can change active sites and change their conformation in order to fit their substrates better. It's not that they always look like that. You have to induce them to look like that. So here's the thing. The non-competitive inhibitor, this is a really important fact. I want you guys to drill this down. Is that the non-competitive inhibitor, does that show up? Barely? OK. Is that the non-competitive inhibitor binds ES just as well as E. Binds the complex, no writing, just as well as the enzyme. You'll go home, you'll watch it again. You too, at home. I'll find you. You guys remember the first time I did that? You guys remember, who remembers the first lecture I told you not to write? Chapter 2 of biology, when we went over the menstrual cycle. Very complicated diagram. I hope that now that you guys have been studying for a while, you realize how difficult that lecture was and why it's so stupid to put it so early in the biology book. It's a very dumb placement of that lecture. So here's what happens. No extra substrate can overcome this. So the increase in substrate concentration cannot overcome the inhibition. Dude, it's been 3 hours. 3 hours straight. What's up?
How are you saying that it's binding to the ES when it doesn't bind to ES?
Pause. This doesn't bind to ES.
No, you said the non-NC inhibitor binds to the ES, but it's
No. Hold on. I'm going to explain that in a second. She asked a very good question. This is a vocab thing. Inhibitor binds to an allosteric site. It binds the enzyme substrate complex, the allosteric site of the enzyme substrate complex, just as well as the allosteric site of the enzyme.
Oh, okay.
Do you want me to write that out? The allosteric site of? Will that make sense? OK. Because if you're confused, then someone else is as well. Binds the allosteric site of ES and E equally. The allosteric site of both ES and E, it binds them equally. No extra substrate can overcome the inhibition, right? This actually decreases Vmax. We'll take a look at why. And the Km, interestingly enough, we'll talk about why in just 1 second, no change. No change in Km. Pause. We have E plus S, ES, E plus P. The inhibitor can bind both E and ES. This will make EI and ESI. Anyone else have a red marker by any chance? Why does this not change? Throw it. Why does this not change? If you have more, throw them. Just throw them. It's fine. Why does this not change the? You almost landed it. That was really impressive. Why does this not change the Km? Let's talk about why it changes the Vmax first. The Vmax is entirely dependent upon what? K1, K-1, Kcat. The Vmax is actually entirely dependent upon Kcat, because remember, the enzyme concentration doesn't change. Kcat is the forward reaction that goes from ES to E plus P. So what's going to be the rate? It's going to be Kcat times ES, right? Now here's the thing. The forward reaction, since that's dependent upon the ES, and the ES is getting inhibited, doesn't that drop the ES? Doesn't that drop the rate? Doesn't that drop the forward reaction? Which means that the Vmax goes down. The disappearance of ES is why the Vmax goes down. Make sense?
Is this why we have a differentiation between Km and catalytic?
Yes. This is the reason that we have a difference between Km and catalytic efficiency. Very good. Is because the Kcat can be affected without the Km. Without a change to the Km. Why isn't the Km affected? Yusuf, you said that the formation of ES and the dissociation is what matters when you're connecting over into the Km. Take a look at Km. K-1 is affected because that has to do with the ES. K1 is affected because that has to do with the formation and you're disappearing from the E. And Kcat is affected. Everything inside of Km is affected by the same amount, so it doesn't change. The affinity remains the same because the appearance and disappearance of everything on the board from the perspective of the Km, nothing's changed. If everyone goes down, everyone remained the same. Does that make sense? Time to take another bite of my burrito. I'm not eating on camera, dude. It's not happening.
Hey, Meta. Cold Claire. Hello. I do have some gossip to tell you. Are you still in New York?
With Sours by Extra, every chew is sour.
You guys can eat too. Let me make these a little smaller. I've said it before and I've said it again. UTD is a jahil place. This place is weird.
Was Brooklyn College just not as good?
Brooklyn College was not this weird. Brooklyn College was full of weirdos, and I was a philosophy major, so there were a lot of blue-haired people. No offense. But, you know, Brooklyn College is pretty weird. Was I one of them? Was I? Yes. So the Km is not affected, right? So both of our graphs will actually sprout from the same place. Yeah? And then if we have a decrease in the Vmax, which way is it going to go? It's going to go up. Because remember, that's 1 over Vmax, right? Oh, man. All right. 1 over Vmax. 1 over Vmax I. Sense? Matters when you're Kcat is the forward reaction that goes from ES to E plus P. So what's going to be the rate? It's going to be Kcat times ES, right? Now here's the thing. The forward reaction, since that's dependent upon the ES, and the ES is getting inhibited, doesn't that drop the ES? Doesn't that drop the rate? Doesn't that drop the forward reaction? Which means that the Vmax goes down. The disappearance of ES is why the Vmax goes down. Make sense?
Is this why we have a differentiation between Km and catalytic?
Yes. This is the reason that we have a difference between Km and catalytic efficiency. Very good. Is because the Kcat can be affected without the Km. Without a change to the Km. Why isn't the Km affected? Yusuf, you said that the formation of ES and the dissociation is what matters when you're connecting over into the Km. Take a look at Km. K-1 is affected because that has to do with the ES. K1 is affected, because that has to do with the formation, and you're disappearing from the E. And Kcat is affected. Everything inside of Km is affected by the same amount, so it doesn't change. The affinity just couldn't get it through my head. And that's why when you're on the other side, and you guys start teaching your own classes about the MCAT, whatever it may be, hope you guys see that day, inshallah, you can just use your notes. You don't remember jack. Although I could draw the entire metabolic map from memory if I wanted to. Might make a mistake or two. 3. I remember one time, there was a group of students who came like after my lecture was done at Brooklyn College, and they just wanted to say hi and like talk about, like I was a great help and blah, blah, and this is right when my channel started. I hardly even had like maybe 1,500, 2,000 subscribers. Maybe, even if that. And they sat there and they were just like, hey, we have a question for you. I was like, yeah, sure. They're like, can you teach us about the RAS? And I was like, yeah, do you want me to use the board? Because like I'd already wiped the board, we like packed up for the day. They're like, no, just tell us. So they sat down and I just like dictated it to them. And I just went and went and went and went and went through the whole thing. And they were just sitting there like, and I was like, I'm gonna use the board. Uncompetitive inhibition. Uncompetitive inhibition is when what happens, guys? This actually binds only to the ES complex. And it prevents the release of substrate. This is very unique. I think of this one, the way I remember this is jail. That's jail. We're going to talk about why. Right? What happens here, and that's the only notes we need. What happens here is that the Vmax goes down.
All right, I think we kind of have that. All right, my fault, guys. Again, this is probably the most interruptions we've had in one day. Okay, 12:28, bro. It's been like 2 hours, bro, and we've only gone through 11 terms. This is actually wicked. Maybe I would have been better off if I would have just gave myself a day off, to be honest with you. What is mixed inhibition? So mixed inhibition. So mixed inhibition, it's basically between like it can be like it's uneven. So it can have preference for the allosteric site of E or the allosteric site of ES. So in non-competitive inhibition, um, it's going to bind equally to E and ES, and that causes there to be a Vmax decrease. And let me look back. I totally forgot to switch the screen, but Vmax decrease and a KM has no change because of the formula for KM. Okay. Let me change the screen capture. All right. Um, so I have the lock and key theory. So lock and key theory says that it's like the enzyme is a lock and the substrate is the key. It has to be a fit. Um, it's like enzymes all have a perfect pH under which it like kind of prefers to function. Um, what is an isomerase? An isomerase catalyzes um, reconfiguration of the bond or rearrangement of bonds in a molecule. It also catalyzes reactions between stereoisomers and constitutional isomers. Kn divided by Vmax. 1 over Vmax. It catalyzes the cleavage of a molecule into two different products via addition of water. Uncompetitive decreases both. Since it decreases both, it sends both of them further. So it creates a okay, all right. It creates a parallel line in which one moves like it moves up vertically and left horizontally. Kcat or K catalytic efficiency is equal to Kcat divided by Km. So we get a decreased activation energy, a an increased reaction rate, very specific, very sensitive to the environment. Um, uh, it's not consumed and it doesn't alter the delta G or the equilibrium constant. There's a competitive inhibitor effect. So the Vmax doesn't change, but the Km increases. What is mixed inhibition? It means that it binds, like a mixed inhibitor is going to bind to both the enzyme substrate complex as well as the free enzyme, but it has different affinities for each one. What that basically means is that the Vmax will always decrease, but it can either increase or decrease the Km, and that's kind of like uh, let me see. I had a chart. I found the chart. It was pretty cool. If I can find it, it is a pretty cool table. See, this is the table I'm talking about. So in both uncompetitive and non-competitive inhibitors, it's the Vmax is decreased. Hold up, bro. Let's try this again. Okay. Um. Okay. So I kind of like these. I kind of like this. So basically, the reason why mixed inhibitors are going to lower Vmax always, but KM can go either way. So, like Yusuf Hassan was saying, when EI or ESI are formed, they basically can't end up getting to the product. So, like it can't create product, and it doesn't matter how much substrate you add. But with KM, if it binds the free enzyme more tightly than the enzyme substrate, it's gonna it's gonna basically drain the pool of like free enzyme. So the enzyme is gonna appear to bind the substrate worse. So KM is gonna increase because affinity is gonna decrease. If it binds the ES complex more tight, then um, it has a higher affinity for the substrate, and the KM is going to decrease because the affinity increases. Okay. Well, all the proteins have a preferred affinity. Right. All the enzymes, they have a preferred affinity. Are a preferred pH. Like most of the enzymes, except for the ones in the digestive system, have a preferred pH of like 7.4. Might have suspend that because it's just like I know that. What is the catalytic efficiency? Kcat divided by Km. Coenzymes are organic no, cofactors versus coenzymes. Uh, this is probably co. It's probably organic molecules. No, because you need an enzyme requires, I think, like the chaperone proteins and stuff to fold properly. Okay, gives us the negative 1/km. They stabilize the transition state. Um, or like the intermediate. organic molecules. They decrease the activation energy, increase reaction rate. Uh, they're not consumed, very sensitive to the environment, very specific, and they do not alter delta G or the equilibrium constant. Sometimes it's the inactive form of an enzyme used like for regulation of that enzyme's products. I guess same thing. If it's inactive, of course it has to be activated. I feel like that's kind of obvious. How do increases in temperature affect enzyme kinetics? This is so like disheartening that it's taken this long. How do increases in temperature affect enzyme kinetics? Um, what is a cooperative enzyme? Okay. I don't even like this definition, bro. I'm just gonna copy this definition and I'm just gonna change the definition that we're using here because okay. When the binding of a substrate to one active site of a multi-subunit enzyme alters its affinity for subsequent substrate molecules. The picture didn't help. The definition didn't help. Like, bro, it's like impossible. This is so annoying. Okay. Yo, 2.5 hours is what we're at. It's literally 1 in the morning, and we started at 10. That's crazy. Um, so okay, we should be at peace now. There shouldn't be any more interruptions. Uh, yeah. What is mixed inhibition? What happens? Okay, so it means that the inhibitor binds differentially to free enzyme and to the enzyme substrate complex. It is going to it's going to always decrease the Vmax, but but um the Km can increase or decrease. Um, for every 10 degrees Celsius increase, the velocity is gonna double, but once it reaches its optimal temperature, after that it's gonna start to it's gonna start to fall off because the protein is gonna start to denature. It means that uh, substrate binding to one of the subunits of a multi-subunit enzyme is going to affect the affinity of the other subunits for their substrate. How do enzymes lower the activation energy? They stabilize the transition state. It's inactive enzyme obviously needs to be needs to be activated. It's used like to regulate the activity of the enzyme. Every enzyme has like its optimal pH. For most enzymes, it's like 7.4, except for the digestive system. There's going to be kcat divided by km. The x-intercept gives us -1. No, wait. The x-intercept, yeah, -1 divided by km. Cofactor and coenzymes are bound by intermolecular forces versus prosthetic group, which is covalently bonded. Is it the same? Is that what I said? Like, that's pretty much what I said. Generally speaking, coenzymes are organic molecules. It is sometimes. Okay, I don't know all these. Ligase, isomerase, lyase, hydrolyase. I think it meant hydrolase, not hydrolyase. A little hot. Wow. Um, bro, how many of these did they spell wrong, bro? Uh, what is kcat? It's the rate at which the enzyme substrate complex gets turned into the product or enzyme end product. starting to get sick and tired of this deck. I'm gonna be honest with you. I feel like it keeps giving me bad definitions. We've been doing this for 2.5 hours. It's in random order, pretty much. I'm gonna look at Jack Sparrow. Hold up. Let's look at Jack Sparrow. I'm gonna look at Jack Sparrow. We might do cars and I don't know, maybe something else, and then we'll call it a day. I don't think we're gonna get through all 50 cards today. I think maybe today is gonna have to be our break day, and then maybe tomorrow I'll do the cards that I realistically should have done today. Um, but to be honest, I might do this whole like I might need to do some research on maybe a better deck. I'm being honest with you. Uh, I thought this was gonna be the deck that I was gonna use, but I don't know. It's like pretty consistently failing me right now. So I don't know if I can rely on it anymore. It's like actually killing me that I continuously keep getting bad definitions for stuff. what we're gonna do is technically have 48 left. Technically, we're gonna change this to 0. We're gonna finish those 7 reviews, and tomorrow we're gonna continue because this is ridiculous. What are the 7 classes of enzymes, or the 6 classes of enzymes? We have ligases, isomerases, lyases, uh, hydrolases, oxidoreductases, and transferases. Mixed inhibition, it binds preferentially to either the free enzyme or the enzyme substrate complex. It The Vmax always decreases, and Km can do either one. The enzyme can be it means that the affinity of another subunit is affected by a substrate binding to one of them. Like if substrate binds to one subunit, the affinity of another subunit's the affinity that another subunit has for its substrate is subsequently affected. It doubles for every 10-degree increase until its optimal temperature, but after its optimal temperature, it starts to fall off. They stabilize the transition state. It's the inactive version of an enzyme. What is the difference between a coenzyme, cofactor, and a prosthetic group? Coenzyme or cofactor is it's it's bound loosely. Ligases, isomerase, lyase, hydrolase, oxidoreductase, and what's it called? Oxidoreductase and transfers. Bro, because it's just like I'm going so incredibly slow. Like it's actually phenomenal. I've never like we have gone all these days. This is gonna be the longest stream. And I literally out of the 50, I did how many what like I did 27, bro. Even out of 50, bro, I did 27. 27 cards in like two like what is that? 160 minutes, 27 cards. That's like atrocious. That's like actually that's I've never seen anything like that, bro. I'm so used to doing like the closed cards that they take forever, but um or they you go through them so fast. All right. let's do some okay. Let's do let's before we do the cars, let's do some research because it's 114. We we need to we need to find some stuff. We need to figure out what the best deck actually is. Because um okay, let me switch the screen. All right. Oh, heat wave exposed. Yeah, I didn't see that. Um, okay. Bro, I can't. Like, are you trying to tell me that, bro, ain't no way. I, I swear this is literally the one that I'm using. I'm using the one off of, I'm using the V2 for sure. I'm like sure that I'm using V2. Captain Hook V1. This is the one that I'm using because I'm not logged in on Firefox. But give me a second. Yep, that's the one I'm using. Okay, so basically, they're all just saying Captain Hook is better. Well, it seems that we have reached our final conclusion. Um, let's go and see. Let's go and see. Um, let's do our cards for the day. Um, I literally just turned the music on just to turn it back off. Okay, hold up. I don't understand this at all. It's probably not A. Okay. I'm gonna stop here because I need to make sure that I'm doing it at least somewhat timed. All right. Let's see. Thank God. Oh my God. That was scary. We can't, yeah, that's what I was thinking. Like warm makeup, it doesn't even sound like it could have been correct. It also, that's why I need to check back because I didn't write in, I didn't write here whether Charlie Chaplin wrote makeup. I mean, war makeup. Um, yeah, I can't say, I didn't see anything about famous. So yeah, I didn't even, this one wasn't even an option for me. So, okay, cool. I feel like they were talking about the ideal face so much, like ideal face of mankind. Like, so there we go. No, this one has to be right. Thank you. Yo, this has to be the worst question I've ever seen. What do you mean? This whole explanation is basically defensive. Like it's not supporting. It's basically saying like yeah, like even though they would disagree, and I understand the question about is talking about critics, but it's basically saying like yeah, like maybe they wouldn't agree with any part, but hey man, it's kind of true. Like face is included in any part. Why in the world would it like would they agree with that? They literally didn't talk about any other part. I feel like in any other question they would have marked that answer choice wrong. We can end the test. I got a 4 out of 5. That's pretty much what everyone got. That was one hard. Yeah. So that was literally that genuinely felt rigged. But okay, whatever. If they say so. All right. Let's see. Let me see if there's anything else that I can do here. Um, I really don't want to. I feel like I'm probably gonna end up just continuing Captain Hook tomorrow. Unfortunately, I'm probably just gonna continue with Captain Hook because hold up, 'cause we're gonna download the Captain Hook Anki deck that doesn't have any updates. We're gonna download that one. just so we can see if it's in order. Because why I'm sitting here doing this, and I just hate how it's organized. Mine an old version of Anki. Let me see. 26.05. Go through the whole. This is the thing that I hate the most about Anki. Like it'll keep all of your data, but it doesn't have an update system. Um, yeah. I'm trying to see. I'm trying to I'm trying to import the original Captain Hook and see if like I just want to see what it says, like how it's organized. Because if this one is organized better, I'm gonna be tight. Nope, it's not. Is there just random? Like what? Why is it random? All right, guys. I think we're done for the day. I think that's enough. I think we've dealt with enough frustration for the day. I think Captain Hook bio, I think I had a very good reason to delay it for this long. Um I really wish that it wasn't 2 in the morning and that I hadn't just spent 4 hours on literally like 20 cards. Like it's actually ridiculous. I don't even know. Maybe it was because there were so many interruptions. I'm not sure, but this is genuinely insane that it took this long. Um, but I hope everyone has a great rest of their night and their morning and their afternoon and your evening. And yeah, bye.